So today we are going to continue with last time's portion, that is photosynthesis. So we are going to concentrate on mechanism of photosynthesis. So when we talk about mechanism of photosynthesis, you have already learned in your lower classes that photosynthesis can be completed in two reactions, light reaction and dark reaction. So by light reaction, what it means is I've already told you about the two pigment systems, PS1 and PS2. So in light reaction, what happens is when light falls on pigment systems, these get excited and release electrons. These electrons go through a series of systems forming ATP and NADPH. These are the main products of light reaction. In addition to this, oxygen is also formed, which is evolved as a byproduct of photosynthesis. Now, ATP and NADPH are again utilized in dark reaction. So, I'll tell you how ATP and NADPH are formed in light reaction. So what happens is, in your light reaction, when light falls on pigment system 1 and pigment system 2, both release an electron. PS2 releases electron to a primary acceptor, which in turn converts or transfers the electron to a series of systems and then finally releases to PS1. At the same time, when light is also incident in PS1, it also releases an electron. This electron combines with ferredoxin, in which NADP is converted to NADPH. Now, the electron lost by PS1 is utilized in formation of NADPH, but this lost electron is compensated from the electron by PS2, and this PS2 leaves an electron hole in itself, while this electron hole is compensated by photolysis of water. By photolysis of water, what happens is water molecule, it splits in the presence of light, producing hydrogen and hydroxyl ions. These hydroxyl ions are highly unstable and they again recombine to form water and electrons. These electrons itself fills up the hole left by emission of electron from the pigment system 2. So these are the series of electron transfers which involves primary acceptor, plastoquinone, cytochrome system and plastocyanin, leading to formation of NADP and NADPH and ATP. ATP is generally formed when electron is transferred from plastoquinone to cytochrome system and from cytochrome system to PS1. So this electron system also involves two types of reactions: cyclic photophosphorylation and non-cyclic photophosphorylation. In non-cyclic, both pigment systems PS1 and PS2 are involved, whereas in cyclic, only PS1 is involved. Now, why is it called cyclic and non-cyclic? Is because in cyclic, the electron lost by PS1 is returned back to itself. But whereas in non-cyclic, the electron lost by PS1 is not returned to itself, neither is the electron lost by PS2 returned to itself. So in order to understand this, just take a look over here. When light falls on PS1, it loses an electron. This electron, it reaches ferrodoxin. So an electron hole is created in PS1. Now this electron hole is filled due to electron loss from PS2. This electron moves through a series of reactions and it reaches PS1. Since electron loss by PS1 is not returned to itself, rather this electron is lost to ferrodoxin. But even though it gets an electron, the electron that is getting it is from PS2. So the electron lost by itself is not returned to it, rather it is getting electron from PS2. But whereas the electron lost by PS2 is not returned to itself, hence this is called non-cyclic photophosphorylation. Now the next one, in cyclic photophosphorylation, what happens is when light falls on PS1, it loses an electron. This electron, it reaches ferrodoxin. The ferrodoxin also returns the electron back to the cytochrome system, and from the cytochrome system to plastocyanin and PS1. Since the electron lost by PS1 is returned back to itself, this is called cyclic photophosphorylation. So generally in your light reaction, due to your electron transport system and photophosphorylation, what we're getting is ATP and NADPH. These two products are utilized in dark reaction. Dark reaction was discovered by Blackman, also known as Blackman reaction. So in dark reaction, Kelvin cycle is involved, which fixes carbon dioxide to glucose. So the Kelvin cycle is named after its discoverer Kelvin. It is also known as C3 cycle. Why is it called C3 cycle? Is because in this particular cycle, the first product formed is a 3-carbon compound called phosphoglyceric acid. Because this compound is a 3-carbon compound, it is also known as C3 cycle. So generally in Kelvin cycle, there are 3 steps involved. The first one is carboxylation. Second one is glycolytic reversal. And the third one is Rubp regeneration. So the first step is carboxylation. By carboxylation, what happens is carbon dioxide combines with ribulose biphosphate to form a 6-carbon compound. This 6-carbon is highly unstable, so it gets converted into phosphoglyceric acid. Glycolytic reversal, it is just the reversal of glycolysis. In this particular step, what happens is the PJ that is formed is converted into diphosphoglyceric acid. This diphosphoglyceric acid is again further converted into phosphoglyceraldehyde, which remains in equilibrium with its isomer, that is phosphodihydroxyacetone. So these two products are formed in glycolytic reversal. In RUBP regeneration, what happens is RUBP is a 5-carbon compound This compound has to be regenerated so that again it can combine with atmospheric carbon dioxide. So in RBP generation, there are a series of reactions involved. So in the series of reaction involved, the main products formed are fructose 1,6-diphosphate, which is a 6-carbon compound, and cereoheptulose 1,7-diphosphate compound. This is a 7-carbon, this is a 6-carbon compound. These products lose phosphate, one molecule of phosphate. So these are converted into fructose 6-phosphate and cereoheptulose diphosphate. Next, what happens is these products are again converted into a 4-carbon compound erythrose phosphate, ribose phosphate, and xylulose phosphate. Erythrose phosphate is a 4-carbon compound, and ribose phosphate and xylulose phosphate are 5-carbon compounds. Now, these 5-carbon compounds are again undergo a series of reactions and conversions, finally producing ribulose by phosphate. So these phosphate again combines with atmospheric carbon dioxide. So this cycle keeps on continuing. So finally, when REBP is regenerated, we also get a molecule of glucose. Now, in addition to the C3 cycle, certain plants are also capable of C4 cycle. By C4 cycle, what happens is, in this particular case, the first compound that is formed is a 4-carbon compound called oxaloacetate. So what happens is, atmospheric carbon dioxide combines with phosphoenol pyruvate, forming oxaloacetate. This oxaloacetate is converted into malate. This malate breaks down into pyruvate and atmospheric carbon dioxide. This carbon dioxide enters the C3 cycle, so carbon dioxide is fixed, producing glucose, but whereas this pyruvate again undergoes a cycle forming phosphoenol pyruvate, which again combines with carbon dioxide. So in C4 cycle, the first product formed is a 4-carbon compound called oxaloacetate. It ultimately undergoes a series of reactions forming pyruvate and carbon dioxide, which the free carbon dioxide again enters the C3 cycle. Now, why a C4 cycle is important is because plants which perform C4 cycle are capable of performing photosynthesis even when the stomata is partially opened. Such plants are very helpful in areas where drought is common. So the next is photorespiration. By photorespiration, what it means is respiration done by tissues which are capable of performing photosynthesis. So generally in photorespiration, you have to remember three organelles: chloroplast, peroxisome, and mitochondria. Photorespiration was a term coined by Dio and Decker. In chloroplast, what happens is the carbon dioxide combines with RUBP oxygenase. Now what happens is RUBP is a compound that is supposed to combine with carbon dioxide and form phosphoglyceric acid according to Kelvin cycle. But what happens is this enzyme can also act as an oxygenase when the oxygen concentration is high and carbon dioxide concentration is low. So the same enzyme can act either for carbon dioxide or oxygen depending on their concentrations. So in this case, when oxygen concentration is high, RUBP functions as an oxygenase. So what happens is carbon dioxide is affected by RUBP oxygenase forming phosphoglycolic acid. This phosphoglycolic acid is converted into glycolic acid. This glycolic acid moves into peroxisomes where it is converted into glycine. Glycine is an amino acid. And amino acids, you all know, are the building blocks of proteins. These glycine can also further enter the mitochondria, in which it is converted into serine. Serine is also an amino acid. So in photorespiration, you have to remember RUBP functions as an oxygenase because oxygen concentration is increasing. And basically three organelles are involved: chloroplast, peroxisome, and mitochondria. So Blackman's law of limiting factors, what it states is when a process is controlled by multiple factors, the factor which is the slowest one controls the entire process. So we all know that photosynthesis is controlled by various factors. Just to name a few, it is either controlled by carbon dioxide, light, or temperature. So taking these three conditions into account, just take a look at this simple graph. What happens is carbon dioxide and temperature are constant. So when you're increasing the light intensity, the photosynthesis increases only up to a certain level. Then the process proceeds at the same rate. Carbon dioxide and temperature becoming constant despite increasing the light, these two become the limiting factors, that is carbon dioxide and temperature. Now, if I increase carbon dioxide concentration, again the process will increase up to a certain level and then will proceed at the same rate. Now I've already increased light and carbon dioxide concentration, but over here temperature becomes the limiting factor. So this is what means the principle of limiting factors. A process controlled by multiple factors in which the factor which is at the slowest controls the entire process. This carbon dioxide intake or oxygen production means the rate of photosynthesis. So in this case, in the first case, light is increased, carbon dioxide and temperature is constant. So the reaction proceeds up to only a certain level and then proceeds at the same rate. Next, I am increasing light intensity and carbon dioxide concentration, but temperature is constant. It will increase only up to a certain level and then proceeds at the same rate. So in the first case, carbon dioxide and temperature is the limiting factor. In this case, temperature becomes the limiting factor. So moving on to the last part of photosynthesis, which you've already done in your lower classes. This means factors which control photosynthesis. So the first and the most common one is carbon dioxide concentration. Your carbon dioxide concentration increases, photosynthesis increases, but only up to a certain level. If a carbon dioxide concentration is more than 0.03%, photosynthesis will decrease. Next is temperature. If your temperature increases, photosynthesis will not increase. If your temperature decreases also, it will not increase. Rather, temperature works best at optimum temperature. So your photosynthesis occurs best at optimum temperatures. Next is oxygen concentration. If your oxygen concentration increases, photosynthesis decreases. This is also known as the Warburg effect. Water, basically if a plant is turgid, then it is able to perform photosynthesis at a constant or rapid rate. So these are all the factors which are controlling your photosynthesis. So we have completed this chapter.